okay? So don't forget the flour has a little bit of protein because you never know what those guys are gonna be asking you. So make sure that you know more than they do. If you prepare well, you do know more than they do. Okay, so I want you to be confident that if you prepare well, it shouldn't be major surprises for you. You're gonna go like this group I just met 2 weeks ago. They go, we, check, 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 check. And that's a good feeling, okay? So that's basically what I recommend that you do. Okay, you start, doing this, a mental review of this. And maybe you put some notes. So here it is. So I'm gonna start, look at this. I'm gonna start with the protein. Okay, look at this. The typical American gets an average. We have not seen this. Okay, we have not seen yet. This is part of Unit 1. Okay, this is part of Unit 1. This is how we know what we eat every day. Okay, and there is a question and the simple quiz about this, and we're gonna go through this once we cover this. This is coming from nutrition. Look at this. The typical American, okay, gets about 15% of the daily caloric intake from protein. About 2/3 of this protein that we get in a day is from animal sources, and the other 1/3 is coming from plants. And I'm already giving you an example of plant protein. What was the example of the plant protein I gave you? What was the example of the plant protein I just gave you 90 seconds ago? Yeah, the flour protein, and it is something that we call gluten. So what is gluten? Gluten is a combination of proteins. And I'm going to prepare like a gluten presentation for my own sake. So what is gluten? So I can show you guys that, in case that you are interested. Because, it's almost like, well, gluten is a protein. It's almost like it's a singular. It's almost like chymotrypsin or trypsin or ATP synthase. Actually, gluten is a combination of different proteins. And they are named for those proteins. One of these proteins is called prolamine. You don't have to write this down. Yes, I was reading about that yesterday. Because there is a review in one of the latest issues of Scientific American about autoimmune diseases. I didn't realize how many there are. Close to 80 autoimmune diseases. So one of them is that, what are the people who suffer from gluten sometimes? What is the disease called? Celiac disease. And it can cause, it's a major problem for some people. You see the finger-like enterocytes, which are the ones that are going to be absorbing the nutrients. And because of that celiac disease, you know what happened? We auto-digest all this. It's almost like sometimes guns in bingo remove our fingers. Oops, chop my fingers. Instead of 4 or 5, I only have 2. So it's a very serious, condition. And you can see right there. So look at this pizza, right? So you can see the digestion starts in the stomach, right? Starts in the stomach, mostly by pexin. But the low acidic condition can also partially, digest some protein. Digestion is completed in the small intestine via digestive enzyme from the pancreas. You remember when you went in biochemistry 1 and biochemistry 2, the first 2 weeks of classes? There I asked so many questions to you. For instance, we were reviewing Unit 1, which is a review of biochemistry 2, Unit 1 and Unit 6. And one of the science in one presentation was chymotrypsin. And I asked my students, okay, have we ever talked about chymotrypsin? Or some student went like this, the typical episode, look at me, right? And of course, no one, there was a student, he said, what is a digestive enzyme? He said, oh yeah, it's a digestive enzyme for starch. Then the student go, not for protein. You see what I do? We are doctors, we're trying to be doctors. What is the meaning of doctor? Did I tell you what is the meaning of doctor? What is the meaning of doctor?
Know it all? Oh.
Know everything.
Yeah. Well, you can say anything, know it all. It's shorter and sweeter for me. Make sure that you know everything, you know it all. Okay, you can see right there. Okay. All digestive enzymes are hydrolases. You see that? Because basically that's what is digestion? It's a hydrolytic
We
process.
just went over it the other day.
Where essentially what we do is we break the proteins into amino acids, we break the polysaccharides into simple sugars, right? We break the lipids, the main dietary lipids, and we remove the fatty acids, in which you can see. And this, most of these enzymes are secreted from the pancreas, right? Mostly from the pancreas, because there is a gastrocytogen in active form. So that's why most of the questions that you're gonna get on board are related to that process. What process? Digestion. So that's why it's a good exercise to start buying pizza, which is a good idea as always to buy pizza, regardless of the time. Okay? And you can see it. And here it is. What do we do? Proteins plus hydrochloric acid, we generate. What is another name for oligopeptides? You remove the oligo. You're talking about the same. Peptides, oligopeptides, the same. So don't. What is oligopeptide? Roger never told us about oligopeptide. Okay? And you can see, look at this. What happened? Look at this. This oligopeptide, look at this. What is this saying? Look at this. The oligopeptides, once they reach the small intestine, which is this tube over here, right? They're going to stimulate, this is what it means. They're going to stimulate, right, the pancreas, because this is a secretin, this is a hormone, a pancreatic hormone. That's why they painted in this tube. This is a mostly an intestinal hormone. And we're gonna see this in nutrition again, right? It's stimulating the pancreas. This is what it means, stimulate the small intestine to secrete secretin, and the main target for secretin is the pancreas. So what is going to be the effect of secretin on the pancreas? It's written in the presentation. The stimulation, the secretion of bicarbonate. Okay? And this is one of the things that we get bicarbonate. Why do we need bicarbonate from the pancreas? Why it's essential that we generate?
To neutralize?
To neutralize what?
Acid.
Yeah, we need to neutralize, to bring, because the contents in the stomach, they are very acidic, typically the typical pH. And maybe put the pH somewhere in there so you have an idea on a healthy individual. We have, oh, they took away that. Oh, here it is. Oh man, I like this. Typical pH is about 1 and 2. See that? What is the concentration of protons if the pH is between 1 and 2? Anybody? I like that. Low. Yeah. Actually, it's high. Actually.
It's whatever equals 14, so.
Yeah,
That's the concentration of proton.
I tried.
You made me laugh though. Okay.
It's funny.
So what is the concentration of proton? Why do I write this down? You never know what those guys are gonna ask you.
Yeah.
Instead of saying the typical pH in the stomach is between 1 and 2, they might say, what is the typical concentration of protons in the stomach of a healthy individual? See that? It's almost like sometimes they ask you about the vitamins, sometimes they ask you about the coenzyme. So what do?
No both.
You know both. that's basically what they give you the pH. Remember, 1 molar give you what? See that? And then look at this. Look at this. The initial, look at it. The initial digestion process, almost like the peptides. This peptide. You can see the acidity is going to stimulate this called cystokinin from the small intestine. And there are two targets in contrast to secretin. See, there are two targets. One is the pancreas to stimulate the secretion of zymogen. The other one is the gallbladder. And the gallbladder is to stimulate the, you can see, what is the gallbladder is going to secrete? Bio, very good, very good. So this is written hopefully in the next slide, you can see right there. Look at this. Secretin called cystokinin, CCK, controls secretions essential for digestion. The low pH of the stomach contains stimulate intestinal cells to release secretin. Okay, and you can see the peptides, though the presence of those peptides in the small intestine is going to stimulate the secretion of the intestinal hormone, which is called CCK. CCK stimulates biosecretion, okay, from the gallbladder to support lipid digestion, okay. You know, gallbladder removal is one of the most common surgeries in the United States. Okay, it's almost like they are close to, almost one every year, every day. Gallbladder removal. Okay. Why do people have the gallbladder removal? What is the condition?
Gallstones.
Very good. Do you hear that?
Mm-hmm.
Okay.
Yep.
Okay. There is also something associated with disease. Make sure that you know the name of the disease. Remember, that's what doctors do, right?
Cholelithiasis.
I don't know, maybe that's
Oh.
another name. I'm not familiar with the name.
That's cholestasis.
Just because I'm not familiar, doesn't mean that's part of my ignorance. So, and this is a little bit, this is the gastric and pancreatic zymogens. And zymogens are named two ways. We either add GEM at the end, right? We either add GEM or we add PRO in front. You see that? Look at this. This is how we know we are in the presence of zymogen. You see that?
Mm.
The other one is adding PRO at the end. And that basically means that they need to, in order to activation, we need to remove that. We need to remove that.
Wow, that makes sense.
And here is a nice little graph I show you in Biochemistry 1. See that? The big kahuna of zymogen activation and for make sure that you know this. We are not going to review this. Zymogen activation. Make a note on your notes.
Please. Bitch. Puppy.
There are two important processes. Is I'm gonna say protein, dietary protein, and lipids. This is one process where zymogen activation. The other one is plaque coagulation. We are not going to discuss plaque coagulation, just in case that you get a question. Okay? The process is similar. We partially hydrolyze some blood proteins without that partial hydrolysis of black protein, there is no black clotting. Okay? And here it is. You can see the trypsin, look at this. Trypsin, which is gone, all these enzymes are pancreatic, except for this guy, that's what I say from this. So this is coming from the small intestine. So this enzyme is secreted when we eat, typically with the purpose to initiate the activation of trypsinogen into trypsin, right? Trypsin cannot start their own activation because it's not active. You see that this is not active. So we need this to start, and once we generate a little bit of active trypsin is going to complete their own activation. That process is called autolysis or autoactivation. Board people like terminology, so when you see autolysis or autoactivation and the process of digestion, what does that mean? They might ask you a question about that. Okay? Once we activate trypsin is going to complete the activation, it's going to activate everybody else. You see that? And this is the table. I'm still the table guy. You can see this is the table. And what is it here? This is the zymogen. And all these are secreted as zymogen. However, these aminopeptidases and these enteropeptidases, they are not zymogens. They are, for instance, this is membrane bound. I'm going to show you a graph. What is an aminopeptidase versus a carboxypeptidase? Look at this. Because they are carboxypeptidases and they are aminopeptidases. So what's the difference between one and the other? They typically, this enzyme typically work on tripeptides or dipeptides. Because eventually, because of the effect of this, all these enzymes, let me put a clean page in there so you can see better. So what is an aminopeptidase versus a carboxypeptidase? Think about tripeptide. I'm going to put a tripeptide in there. I'm going to complete the story over here so I can review everything, the structural aspects. This is what a typically tripeptide looks like. And this is just to remind our group that they are always in the transposition. So we are reviewing a few things right here. They are never like this unless they are two proteins next to each other. So we always have the R groups in the transposition. So there is room for the R groups, okay? So where is an aminopeptidase? An aminopeptidase is not specific for any particular amino acid. An aminopeptidase, what it's going to do is going to target the amino end. Remember, this is the amino end or terminal. This is amino end. And this is called the CN or carboxylate. This is the CN or carboxylate. And so an aminopeptidase does not target a particular amino acid. It's going to target the first amino acid regardless of the amino acid. It's going to target the first amino acid. See that? A carboxypeptidase is the opposite. My PhD professor did a lot of work with these enzymes. A carboxypeptidase is going to break the covalent bond between the first amino acid and the second amino acid from the carboxy end. That's what it's called, a carboxypeptidase. Peptidase means, what is the difference between a protease and a peptidase? Protease works on whole proteins. That's why trypsin is a protease, because it take a whole protein and start chopping it next to lysine and arginine residues. A peptidase is one that works better on peptides. So that's why you see over here, you can see. So all of them are, and that's why these guys are not cymogens, and they are membrane bound. So these are intestinal cells. Look at this, they are intestinal cells. Most of these are the pancreas. The only one that is not pancreatic out of this is pexin, which is in the stomach. And you can see why so many proteins. Why so many enzymes? Because they target different residues. They target different amino acids. You see that? For instance, look at this. Pexin typically, look at this. Typically, I want you to, all these enzymes are CSI enzymes. I don't know if you remember that. I want to make sure that you know. For instance, I'm going to put a protein in here. Here it is. I'm going to put an amino acid over here. I'm going to put lysine over here, and then I'm going to say this is amino acid. Fine, we go on. This is a protein. You see that? For instance, this is lysine. This is the enzyme or lysine. This is the CSI. We are focused on this amino acid. This is this enzyme. This is the CSI. All these enzymes, they target the CSI amino acids. You see that? That's what it means. Okay? Just in case that you get a very, will be a picky question, but you have to be prepared. You want to be over-prepared. Okay? So that's basically, for instance, what does it mean? For instance, if this guy, for instance, I'm going to paint it red. Think about this if we have phenylalanine in here. You see that? If this amino acid 4 is phenylalanine because it's going to be hydrolyzed by pexin. You see that? That's what it means. So pexin is going to come right here and it's going to break right here. This is going to be pexin. And that's exactly, for instance, lysine and arginine, that would be the case of trypsin. You can see what is aromatic amino acid. For instance, chymotrypsin prefers to break next to tryptophan. Make sure that you know what are the aromatic amino acids. Tryptophan, phenylalanine, and tyrosine. Okay, look at this. The proelastase prefer small amino acid. You can see the carboxypeptidase and the aminopeptidase. We already explained this. And this enteropeptidase does not target dietary protein. Remember that the role of this enzyme is to initiate the activation of trypsinogen. Remember that? That's what it's doing. So this does not target. And that's why the target for this is trypsin. And it's going to break next to the CSI or lysine 5. We studied that. I don't think they're going to ask you with that specificity. I always give you more than you want to be asked. And here it is. These are the aminopeptidases. For instance, this is the aminopeptidase. It's this yellow protein right here that is embedded in the membrane. You can see right here. Look at this. Think about this. For instance, if this is a tripeptide, what this enzyme is going to do is going to remove a single amino acid on the amino end. So if we go from a tri, it's going to generate a di plus a single amino acid. That's why the single amino acid is going to be internalized with the free amino acid right here. Think about all those enzymes. Make sure that you can name these enzymes. The pexin, the chymotrypsin, the trypsin, and the elastase, and all. And then we're going to generate free amino acid. And there is a transporter. You can see sodium amino acid importer. Okay? And then, for instance, if this is a dipeptide, if this is a dipeptide, bingo, we're gonna generate 2 amino acids, and both amino acids are gonna be internalized with the green protein. But it's a tri, then we're gonna generate, if a tri, we remove one,
It's dye.
the di is gonna be internalized this. Look at this, this is tri and dipeptide, these are 3 amino acids. You have to understand what this is in here. And what happened over here, some of these amino acids are gonna be used by the intestinal cells to do what? Why intestinal cell need amino acids?
Grow and rebuild.
Yeah, rebuild to make their own protein. But some of the most of the amino acids are gonna be, and you can see, gonna be, to live via portal vein. See that? And this, we have proteases, we have peptidases that they're going to convert this into free amino acids. You see that, free amino acids? Okay? This is how the liver is gonna get a new supply. I'm gonna show you that essentially the protein requirement, everybody is in a high protein diet. And it's typical, you can see, I show you that about 15% of the calories in the typical American coming from protein. That's about twice as much protein as we need. I'm gonna show you the requirement for protein is that we supply about 10% of the calories from protein. So we consuming as twice as much. And people go and say, I'm gonna go into a high protein diet. We already are in a high protein diet. So we are fortunate there is plenty of protein in here. And there is nothing wrong with the amount of protein that we are consuming. Okay? And we're gonna see in nutrition that this is a challenge for some countries. Okay? And you can see this is gonna go to the portal vein. So this is a nice little review about, and what do we do with amino acids? Well, we're gonna see there is some pathway about amino acids and protein, and you can see right there. So let's talk about the transporters, because that could be, look at this, this is called secondary active transport. What is secondary active transport? Secondary active transport means that we don't use it, because there is another active transport which is called primary, and that's where the ATP is involved. Secondary active transport in human is done with sodium. That's why sodium is the highest in the blood. What is sodium doing that transport? Secondary active transport. Okay? For instance, bacteria does transport with protons. We don't do transport with protons. The transport that we do is typically with sodium. Okay, and that's why sodium is so elevated in the blood. And you can see that. Basically what we do, we push the amino acids with the high concentration of sodium. The sodium concentration in the blood is equivalent to that in the gut. Okay, we're gonna see this in the mineral units and nutrition again. And why there are different transporters? That depends on the nature of the amino acid. Remember, the nature of the amino acid is dictated by their group. For instance, probably the glutamate and aspartate, they are transported by the same carrier. Why those two amino acids, what do they have in common, aspartate and glutamate?
Yes,
What do you say?
no, I said it.
Yeah, they are acidic.
Sorry.
They are acidic amino acids. So, if we do that, there might be 4 or 5 families of this transport. But in reality, when you read the literature, they say that there are between 7 and 8 transport for amino acids. So that probably means that maybe those amino acids, for instance, polar uncharged, which is one of the largest of the families of the hydrophobic amino acids. Because if you include, maybe there is one family for the aromatic amino acid, even though 2 of the aromatic amino acids are classified also as non-polar. And in one of the hydrophobic, one of the aromatic amino acids is classified as polar uncharged. So maybe when the families are too big, maybe there is more than one transporter for the same family. Okay, and that could happen for the polar uncharged and for the hydrophobic amino acid. And this is what we do. There is always a pool of amino acids in all cells. Part of that pool is generated by this intracellular protein breakdown. This is called protein turnover. So what is protein turnover means? Is that what it is?
Put an R at the end.
There.
Just put an R at the end.
Okay. That's it. Turnover. And you can see right protein turnover, and you can see right here. Okay, protein turnover, and you can see right here. Well, this is something that happens. You know, the only cell that maybe doesn't have protein turnover is which cells?
Protein turnover.
The non-protein ones?
Which cells do not have the capacity of protein turnover? Because that means we degrade protein and we make proteins. So what cells cannot make proteins?
Is it the ornament and the sexual?
The brain makes more protein than many other cells. Red blood cells.
Oh.
There are no ribosomes, right? Elementary, Mr. Waxon. And you can see, I'm gonna show you this. The first thing that we do when we catabolize amino acid, we remove, these are two reactions that we do. There are two reactions that we're gonna start later. Okay, most of the ammonia is converted into urea in the liver. So what happens when we convert ammonia into urea? Chemically, what happens? I'm gonna show you the structure of, I'm gonna show you the structure of urea. So you can see what's happened chemically. This is urea, this is ammonia. What changes?
You lost the, you gained in oxygen.
Okay. Actually, the effect is the charge.
Oh.
Too many charges because too much ammonia is gonna be put, it's gonna produce an osmotic imbalance in the brain particularly, and that's the reason why, you know, too much ammonia in the blood is very toxic, particularly for the brain. So this is toxic in high concentration. This is not toxic. So when you run into your liver, you say, thank you. And when you urinate today, you say, there goes my nitrogen. You see that? Because that's where the nitrogen goes. And then we eat to replace the nitrogen. So how much nitrogen do we have to replace? About 10% of the calories. That's pretty much it. I'm gonna show you this in nutrition. So we don't have any problem replacing the nitrogen that we use because we have a high protein diet. And people go, if I'm gonna go into a high protein diet, which basically means that, you know, sometimes they double what they already getting. And that could be potentially. If you go too high, that could be. It's like everything. Okay, and you can see what happened with the carbon skeleton, which is the part that is left after we remove, mostly converted to glucose via gluconeogenesis. We're gonna see that in one of the sessions. So let's go and digest now the crust and the pizza, which is in my book is the best part, right? And you can see this is coming also for nutrition, and we have not studied this yet. The typical American gets on average about half, I'm gonna say half, about 52% on the calories from carbohydrates. Half of them are called simple, okay, and half of them are complex. We're gonna see that. We haven't studied alcohol yet in Unit 1. And you can see it right there. Most dietary carbohydrates are coming from plant except for lactose. And we only get lactose if we get into the dairy. We haven't studied the food groups. We're going to study the food groups in Unit 2. One food group is called, we used to call it the milk, now it's called the dairy. Because some people didn't realize that actually cheeses are coming from milk. Can you believe it?
What?
You never went to a farm, right? You never spend a summer in the, with your family, making cheeses. It was a lot of fun. It's one of the good memories I have of having families, that they have farms, so they were managers in farm and stuff. I will finish elementary school and the following day I was in the farm with my cousins. Okay, I don't know how I am alive because we did some crazy stuff in those farms. Okay, then you can see the main dietary carbohydrates are starch, sucrose, lactose, glucose, fructose, and cellulose. But remember, there are two guys that they do not need digestion. Look at this. These are monosaccharides. See, these are monosaccharides. And these are the only two monosaccharides that are present in significant amounts in many food. For instance, fruit, veggies, nuts. They have these two. Okay. The only dietary source of galactose is lactose. So if we don't get into the dairy, we are not going to be consuming any galactose. Mannose is mostly associated with glycoproteins, so it's not in free form. And it starts in the mouth. We have an enzyme in the saliva, which is called alpha-amylase, salivary alpha-amylase. Okay, it's also called known this. There are two isozymes of alpha-amylase. This is the only one that's called this name. When you see names like this, those old guys, these are classical names. They like classical stuff that's been around. It's almost like, I don't know, it's like Mozart, it's all, if you are into whatever music is out there, So remember, you cannot forget about Mozart. But if you don't know anything about Mozart, you might fail. They ask you a question about Mozart and you didn't know. Who was this guy? Maybe invented the microscope. No, that was another guy. See that? So that's what it means, it's classical stuff.
Yep. Robert Cook.
So basically, yes, and actually what alpha-amylase is gonna do with the starch is gonna generate pieces. Those pieces generated by alpha-amylase in the saliva, in the mouth, they are no longer substrate for alpha-amylase. Okay? It's almost like alpha-amylase comes to start chopping, the two polymers in starch. And it's gonna generate pieces. The main piece is called maltose. Okay, but it's also generate maltotriose and isomaltose. Maltotriose, glucose, and maltotriose are no longer substrate for alpha-amylase, and that's why we need 3 other enzymes to complete the digestion. There is a little story, and you can see, look at this. All these 4 enzymes, look at this, all these 4 enzymes, it's almost like we do this starch, right? And then we generate pieces. So we generate maltose, we generate isomaltose, and we generate maltotriose. Maltotriose. And this is what alpha-amylase is going to do. Alpha-amylase is going to come over here and it's going to generate these pieces. Now we need 3 extra enzymes to break these pieces. See that? And that's why now we want to need maltose. So maltase is going to come over here, right? And then you have glucoamylase, which is going to come over here, and then you have isomaltase is going to come over here. I know it's messy, but you get the message. So we need 4 enzymes to convert the starch polymers into glucose. So all these enzymes, and you can see all these enzymes, mostly the enzymes that digest carbohydrate are mostly intestinal, which is in contrast to dietary protein, which are mostly pancreatic. The only one that is not intestinal is alpha amylase. There is one isocyme in the saliva and there is one in the pancreas, but they essentially do the same. Okay, and that's basically, make sure that you know what are the target, the linkages. And I put lactase in there in red because it's a beta linkage. And humans have difficulties breaking beta linkages. And here is, I like this graph very much. It's similar to the previous graph. Now we generate a monosaccharide, right? This is mostly happens in the small intestine. There is nothing happening in the stomach in regard to carbohydrate digestion. There is a little bit of digestion in the mouth. So here is glucose and galactose is internalized by the same type of transporter that transported the amino acids. You see that? Secondary active transport, you can see right there. Okay, maybe put a note over there, even though it's in the presentation in the next page. So this is going to transport. So here is glucose and galactose. So maybe put another arrow, put this G and the other one, put another arrow for galactose. Okay, so this protein, this sodium in galactose. That's why I put galactose in there. So again, this intestinal cell is going to use some of the sugar for energy. Some of this glucose, some of these sugars are going to be fully oxidized for energy by the intestinal cell. So forget about that. After we eat, all cells use glucose for energy. All cells, there are no exceptions. And the reason is, remember, we are given two choices of energy, fat and carbohydrate. Cells know that we cannot store a lot of the carbohydrates. So we use the fuel that we cannot store. We can store a little bit of glucose in the form of glycogen, but it's limited mostly to the liver cells and to the muscle cell in some proportion to the brain cell. And then you can see right here, you can see that this is, look at this transporter is different from this transporter. This is active, this is passive, this is part of the GLUT family. So you can see that this is GLUT2. Okay. The GLUT families are passive transporters. This is another part where they might get you. And then what happened with the other main dietary carbohydrate, fructose? Fructose is prominent. If you get into fruit, if you get into veggies, and you get into nuts, they have a little bit of fructose. So you have to deal with that. And there is another member of the GLUT family, which is GLUT5. GLUT5 is mostly a fructose transporter, and GLUT5 is found in many cells, including the small intestine for absorption and release. And again, this fructose can also be oxidized for energy. And remember, some might be, see that? And all this is gonna go to the,
Liver.
to the portal vein. And portal vein is gonna stay this straight to the liver. And here is, this is a, so this is basically, for instance, the intestinal cell, and all cells have GLUT2 and GLUT1. I mean GLUT1 and 3. Okay? There is also the sodium potassium pump, because every time that we bring a molecule of glucose or galactose, we bring 2 molecules of sodium. I'm going to go back to the graph. So we have to do the equilibration of that. See that? Look at this. Every time that we bring a molecule of glucose or galactose, we take 2 molecules of sodium. And that's why most cell has these pumps, sodium potassium pumps. Because eventually we have to equilibrate this. And this is the point about this. For instance, this is very prominent in the brain. The brain uses a lot of energy after delivery. I'm gonna show you. Okay, I think it's in the next unit or one of the units. After the liver, there is no organ that uses more energy than the brain. And most of the energy is done for this electrogenic transport between sodium and potassium. This is what produces the wiring on the brain. Without this wiring on the brain, there is no communication. And this is part of the Alzheimer's process. Those N-terminals, they can now connect to each other because there is accumulation of beta amyloid proteins. And that's where they are. We start losing our memory. So this is a nice little review. And here is the transporter. They are carriers. Okay? Transporter, they are carriers, so they are channels. Carriers are all typically for organic molecules. Channels are for ions. Okay? And they are based on we go from low to high or from high to low. There is the passive and the active. So this, I put that review over there. And that means the directions. There are uniporters. For instance, the GLUT families are uniporters. What is the meaning of that uniporter? For instance, they transport a single molecule. For instance, look, this is a uniporter. Look at this. You see the transport the glucose or galactose in one direction. That's what a uniporter. This is an antiporter. What is an antiporter? It's one that transports the 2 things at the same time, but in opposite
Different directions.
directions. So make sure that you are. And that's what I put a little bit of review over there, so you can get into that. Okay? And I don't have time to go through this, but we're gonna see the same table in nutrition.
Okay. Thank you.